Good afternoon. I'm going to get started since we are a little late. Welcome to the Department of Medicine Grand Rounds. Today's conference is hosted through the Division of Nephrology and Hypertension in recognition of the former chief of our division, Jacques Bourgonnier. Dr. Bourgonnier was chief of the nephrology division from the late 1970s until about 2000. And during those several decades, he really created a foundation upon which the current division's success was founded. He, during his long career, he was a physician scientist for many years, sort of evolved into a clinician, both of which he did extremely well. And importantly, he created a lot of opportunities for junior faculty at this school, many of whom have progressed to become senior faculty here, including myself. In the end, I would say that those who knew him, and many of those obviously who did not know him, remember him as a very kind, humble, and respectful gentleman who was extremely dedicated to the mission of academic medicine and really committed to the Division of Nephrology and Hypertension here at UM. Upon his passing several years ago, his friends, colleagues, and family came together, created an endowment that allows this lecture to be given every year. His wife, Chantel, could not travel from Europe to be here today. She is hopefully on the Zoom to be with us for this conference. And I'm very excited for us to introduce today's guest speaker. Thank you very much.
Thank you so much, Dr. Roth. So I'm given the task today to introduce our Grand Rounds speaker. Today, I am honored to introduce Dr. Olga Troyanskaya. Dr. Troyanskaya is a professor of the Department of Computer Science and Louis Ziegler Institute for Integrative Genomics at Princeton University. She completed her bachelor in computer science and biology in the University of Richmond, Virginia, followed by a PhD in biomedical informatics at Stanford University. Dr. Troyanskaya is the Deputy Director for Genomics at the Flatiron Institute Simons Foundation in New York. In addition to her duties, Dr. Troyanskaya is part of the National Advisory Council for Human Genome Research, as well as Board of Directors member in the International Society for Computer Biology. She's part of multiple scientific advisory boards like Paris Life and Goldfinch Bio. She was chair of this scientific advisory board group of the Genome Space Consortium between 2012 and 2016. She is currently part of the advisory board. Dr. Troyanskaya is the recipient of multiple accolades and awards, like the Ira Herzowitz Award of Genetic Society of America for outstanding contribution in the field of research, as well as the Overton Prize of the International Society of Computational Biology, and the TR35 Award of the MIT Technology Review magazine as one of the top technology innovators in the world under the age of 35. She is an international renowned researcher with over 150 publications, and she has received several impactful grants funded by the NIH NCA for her work on genomics and precision medicine. Dr. Troyanskaya.
Thank you so much for the kind intro. I think the mic is working. So I'm really glad to be here and very excited to speak to this clinical audience. If you guys have, I know usually everyone is quiet until the end, but if you have questions that like something totally doesn't make sense, it probably isn't making sense to everyone else. So do tell me those questions ahead of time, please. All right, so I'm going to talk about machine learning approaches for precision medicine. And what I'm really hoping to tell you about is what can we actually do and how can we actually understand all of these links between your patient's genome and clinical outcomes, right? So, one most basic question is, how do these variants in the patient's genomes actually impact transcriptional and post-transcriptional gene regulation, right? So there's obvious things like, oh, there's a variant and it completely knocks out a function of a protein. There's a premature stop codon, the protein isn't made, and it's a protein that you know is clinically important. Well, then we know what to do. But what do we do for everything else, right? Like, what if a variant shuts down the expression of a protein? So now the protein theoretically could be very functional, but it's not made at all, right? So how do we understand this? And then how can we actually go from sequence to actually understand what it means in a particular cell type and tissue context, right? So just understanding that that variant might impact expression of a protein, that's great. But if you are a kidney disease doc, right, and you really care, or you're looking at a patient that potentially has FSGS, you really want to look at podocytes, right? So how can you understand that in a tissue-specific and cell type-specific context? And then finally, maybe it's a variant that's functional, it's biochemically regulating something, it's really affecting expression of a protein, but is it actually causal for disease or causal for potentially the way that the drug effects are happening? All right. And so I'll tell you about 3 methods. Don't worry too much about the particular types of machine learning that it's using, but there are machine learning approaches for predicting epigenetic effects. So that means biochemical effects of how variants are affecting, chromatin states in these patients, right, of variants, and then taking that all the way to regulatory activity. So can we take this A to C mutation in a particular location in your patient and then be able to say that's actually knocking out a liver-specific enhancer, and that might actually be linked to their cholesterolemia, right, effect. And then we can actually go further. So this is whether it's knocking out tissue-specific enhancer, but can we actually predict expression effect? Can we look at a patient's genome? We don't have any expression data for this patient, right? But you're trying to understand, you really care about, this variant, and it looks suspicious because it's next, it's upstream from a gene that isn't potentially involved in the disorder you're studying. Well, can we actually say, based on no other data, that that variant is going to turn off expression of that gene or express that gene a lot higher? And in specific cell type, of course. And then finally, of course, it's not just expression that matters, right? Genes are being spliced. These RNAs are being transported, especially, actually is neurology in medicine? Neurology is in medicine, right? No, okay. Well, some of you might care about neurons in some way, right? Like especially you, or any cell type, right? That has longer shapes, right? Like then actually RNA transport matters. RNA stability matters actually for pretty much any complex disease where you care about, right? Just because you are making the RNA, if it's being degraded immediately, or if it's hanging out too long, that could affect not just your diagnosis, but of course, drug metabolism, et cetera. So being able to understand how variants might affect splicing, transport, and other processing, post-transcriptional processing is also critical. And then finally, actually, I'll tell you how we can, if we have time, I'll tell you about how we can integrate that all in the network context. So how do pathways and cells act? So why is it hard to interpret genomes? So all of these individuals might actually have the same disease, even the same subtype of a disease, but it might be caused by very different variants and very different backgrounds. And of course, the vast majority of research in the past few decades has really focused on sort of the top of this iceberg. We don't have, do we have any way to point? I think this might be, I'm afraid to push it. Let's see. Okay, I don't know. There, I can point like this. The top of this iceberg, right? On the exons, on the protein coding regions of the genes. But of course, 98% of our genomes are actually non-coding, right? So this is all the variants that are affecting whether a gene is turned on or off, what level of protein you're being made or RNA being made, and how that RNA is being processed, right? So if you have a misspliced protein, that protein might not be functional at all, or it might have totally different stability characteristics and wouldn't act in the way that you would expect. And so that's a huge search space, right? That's 98% of 3 billion letters. That's this deep, dark space of regulation. And so that's where we're going to look at. And of course, the space is so large that our standard way of looking, like, what we usually do with genome-wide association studies, where we sequence patients that have the disorder, let's say Alzheimer's, or a particular type, like say FSGS, right? We sequence patients that don't have FSGS, and we'll look for variants that are specific to the FSGS group that are not in the FSGS group. So that has a couple of issues. One is, of course, then you're only looking at common variants that occur in the population very often. And many of these diseases, especially actually in the kidney space, are linked to rare variants, right? Or variants that are, or combinations of variants. And the second problem, of course, is that if you're talking about 98% of the genome, the search space is just so vast that you're not going to see the same thing multiple times, even if you have huge cohorts of potentially even tens of thousands of patients, which we don't actually still have on whole genome studies, right? So potentially I would even say, even if we sequenced our entire population and deeply phenotyped them and had deep clinical information on all of them, we wouldn't have enough power to be able to associate every variant that's important in human disease, drug metabolism, et cetera, so clinically relevant variants to their phenotypes, right? So how do we do it? What we actually need is what all of you have seen, potentially for some of you longer ago, for the residents and fellows more recently, right? In your molecular genetics textbooks, right? The coding, the genetic code where we actually can map variants in the amino acid encoding codons to their outcome on the amino acid that's going to be made. We need that for the regulatory space. So we need something that for any variant can associate it to its outcome. And it turns out that these deep learning models are actually a powerful tool for mapping this, for really being this regulatory code. You've all used deep learning. If you use Siri or Alexa, you use Google image search. I mean, it's everywhere now. For those of you who are sadly not 35 anymore, even close, you probably remember it used to be that if you called the call center and there was an automatic assistant who needed to understand what you're saying, like they couldn't even understand numbers, right? Like you would say one, especially if you have a Russian accent, one, and now you can do free speech, and most of the time it actually understands what you mean. That's all deep learning. So it turns out that it's not a solution for everything in medicine at all. There's big issues with deep learning models that we won't go into it, but it turns out for interpreting genomes, for these sort of sequence type of questions, just like for text, right? Just like for our human speech, it's actually very powerful. And so we've actually proposed one of the first models, and then since then it's been a huge explosive industry of these models. But what I want to tell you about is our most recent approach called SAY. And so what SAY does is for any variant, for any change in the genome, right? If you tell me there's an A in this particular position in the genome, the goal of SAY is to tell you what biochemical effect it has. And that is, let's say it's affecting histone mark at this position in this way. And then what does it actually mean in terms of regulatory activity? It's destroying liver-specific enhancer, for example. So what that means is we actually, and we did this for the whole genome. So it turns out these models are very, very computationally intensive. So we actually go in 100 base pair jumps through the whole genome. So now we have the genome tiled with 30 million sequences, 100 base pairs at a time, right? So you look at position 0, you put all the chromosomes in order, you look at position 0, you look at that sequence and so on. And so now your entire genome is covered. You put it in the sequence model, and remember, the sequence model is your regulatory code. Given a sequence, it can predict what is the epigenetic landscape. So, right, every histone mark, so, over 22,000 different histone marks, transcription factor binding sites, whether DNA can be open with that variant there, or if it's closed, right? So is it ready for transcription or not? So that's what it's predicting. And what's really important is these models are not trained with mutational data, right? So they're actually, and if you're interested in how this works, I'm happy to give you intuition later, but it's too probably complicated given the timing. But they're not trained with any mutational data. They're just trained on the reference genome, which means that they can predict effect of any variant. They don't have the statistical issue that genome-wide association studies sort of traditional ways. So we can predict rare variants, or we can predict any, you literally can tell me, change this A to this C, and we can do that. And it has no bias in accuracy for that. And it can do this, so we have this based on this amazing Systrome database, which covers, really a huge amount of public efforts that NIH has funded for finding these cheap-seq profiles. So now we can do about 22,000 different profiles, about 1,300 cell types. There's over 1,000 transcription factors, about 77 histone marks, and they're all quite accurate. So it doesn't matter what AUC is, it's a measure of accuracy that is widely used. Again, I'm happy to tell you, but it's, think of it as accuracy around 97%. So it's pretty good for biochemical experiments across the whole genome, right? So now what's critical is you can actually take this. So this is biochemical effects. But especially for you guys, right? Like I have a feeling that unless you're in a very biochemistry heavy lab, you don't actually care what it means that this A to C change means that this particular histone mark is affected, right? What we actually want to do is at least a regulatory, real functional effect. And so you can actually cluster these marks. And what you see is these classes, so FOXA1, ASR1, AR1, hopefully if any of you are interested at all in, say, breast cancer, this is starting to sound familiar. Or if you look at these marks, actually it turns out this class of these class of sequences all look like liver-specific enhancers, right? And this was done completely in a data-driven way, right? So some of these sequences have been known to be liver-specific enhancers, and others are actually associated with liver-specific enhancers purely by this deep learning analysis. And this is how it works. So I know this is, it's very pretty and looks a little bit complicated. So every dot on this graph is one of those 30 million sequences tiling the genome. And the color of this graph is, the color of that dot is based on that regulatory class, so liver-specific enhancer, right? Or promoter, or a low signal area, which means that basically nothing happening in that sequence, right? Or CTCF. So these are all actually telling us what that part of the genome does. And it's done purely based on this analysis. So that means now, there's your FOXA1, AR1, ESR1 binding, right? So now if you have a variant in your patient in a sequence area that has been associated with that regulatory sequence class, that gives you a mechanism of what that variant might be doing. And same thing, there's the liver intensity enhancer. So all of that we could have done actually based on the data that we used to learn the model, right? So all of those large collections of chromatin profiles. What's critical, why you need the deep learning models? I mean, it actually does help denoising, but the main reason why you need it is this was all done, remember, based on the original human genome, the reference genome. What happens if you have a variant, right? So some of these variants will have no effect on the regulatory class that sequence is in, and others will, for example, like this A to G might actually tell you that the sequence that in its reference allele version was a liver-specific enhancer. Once you have that single nucleotide change in your patient, that now knocked out the liver enhancer activity of the sequence, and now the potentially critical gene is not expressed, and you have issues with liver function, potentially. And you can actually go through all the known disease mutations. So these are the disease mutation. I think there's about 800 known regulatory disease mutations. So these have been clinically, and a lot of times actually, characterized biochemically, or at least in the lab, where there are regulatory mutations that have been experimentally confirmed to be associated with disease, right? So this is not just from a GWAS study. This is actually, shown experimentally, but for many of them, we don't know what the mechanism is. Now you can actually go through them, and that's what we did. For every one of those mutations, we can assign a mechanism. So for example, here's one here. These are polydactyly disorders. And it looks like what's happening is that they're actually affecting the sonic hedgehog pathway by destroying a stem cell specific enhancer that's regulating sonic hedgehog, right? So now you have a mutation that previously has been associated with the disease, and now you have a specific mechanism. And same thing here, there's melanoma with TERT promoter, and retinoblastoma and Cowden disease with other promoter disrupting mutations. Now, this takes you from variants to biochemical effects to regulatory functions. Can we go all the way directly to cell type specific expression? And so this is another project where we're actually going further. So going beyond, right? So I've told you so far, we're predicting these chromatin profiling features with neural network. What if we now take single cell gene expression data, right? So these are data not from disease. This is normal, tissues, single cell expression data. Now, remember what we have the genome, right? For any sequence, we can predict what the chromatin would look like, transcription factor status, et cetera. Now we can actually, so this is in silico, single cell expression is separate, right? Like, so what if we can train a model to actually go all the way from sequence to expression, right? And remember, because we're relying on deep learning models here, what's critical is you can actually take into account not only common variants. So if you try to learn, the obvious way to do it is you have a bunch of patients or healthy individuals, and you take, you make single cell expression data, you sequence their genomes, and you try to associate variants. This way, again, you will only find variants associated to expression change that are common, right? Because you have to see it in many of those people. You're not going to learn anything about most of the genome. In this case, since you're relying on the deep learners, you can actually find this for any variant. And it turns out this actually works. So we've done this for a couple other tissues as well, but actually, surprisingly enough, and to our excitement, kidney was actually the one where we had the most accuracy and the most cell types. So on the left, the 25 for the kidney is the number of cell types. So this is cell type specific expression purely based on sequence. You give me your patient's sequence, we can tell you for each gene what their expression will be approximately, or at least whether that particular variant will up or down regulate the expression. And you can see that across tissues, while there's some variance, right? Like, so the brain, so this is accuracy on the x-axis. So you can see the brain is a little worse and liver is a little worse. Kidney and heart are better. A lot of that is actually just dependent on how much single cell and the quality of the single cell data we have. So as we get more, this will all, I don't think it's some inherent ability to predict brain versus kidney, but so far kidney is doing really well. And so what can we do with this? Well, we can take variants of unknown significance, right? So this is variants that have been reported as clinically important, have been seen or potentially clinically important, right? So they've been sequenced in a CLIA lab because they were suspected of being associated with a particular disorder, and then have fallen. So that's ClinVar, right? That's the database that gathers all of these variants. And the vast majority of them, so about 1% of them then get characterized as pathogenic, and about 44% are these VUSs. So these are variants that, somebody saw them in a patient, they clearly had a suspicion for genetic basis enough to send that to CLIA to sequence. There is a variant. Is it important? Who knows? So we can actually now take this and predict and basically see, does this variant predict an effect in a cell type that's relevant to a disease, right? So in this case, we actually, just by a very conservative threshold, we can actually predict expression disruption based on these VUS variants for 268 of them. And we actually were very interested in PTEN because there were a number of variants of unknown significance reported upstream, so above the start site of the PTEN protein, PTEN gene. And of course, PTEN is known to be important in cancer. We were actually interested in it also because we're interested in neurodevelopmental diseases, and it actually has been implicated in autism as well. So we actually looked at it from the autism perspective in terms of the cell type. So this is, here on the x-axis is just the genome, right? I don't have the numbers, but we're just looking across the genome, right? This is PTEN starts somewhere here, the gene. And these are, each one of those dots is the VUS, one of the variants that has been reported to ClinVar. Obviously, not all in the same patients, right? Somebody reported it at some point. They were all variants of unknown significance. And on the y-axis is the neuronal stem cell specific effect that's predicted by our model, right? So if it's high for this, for example, variant that's the highest, that means that our model says if this variant is there, it's going to change gene expression of PTEN very strongly, right? And you can see that there's a few variants that it predicts change the gene expression, and a bunch of them do nothing. And it turns out then that we can, well, first of all, it turns out that actually out of these models, you can even see what the conserved sequence is that it's disrupting, right? Because you can actually change every possible position to every possible other position and see what the predicted effect is. So you can literally read out the sequence profile, and that's the YY1 binding site of PTEN. So if you're interested in that, but we actually tested this experimentally, of course, not in patients, in tissue culture. But if you look at neuroblastoma cells that are a pretty good substitute for looking at, neuronal stem cells, we actually do see a significant effect of expression disruption, decrease in expression for this protein. And if you look at another cell type, so for example, liver have G2 cells, there is no significant effect. And actually, I'm not showing this for purposes of time, but we've tested all 4 of these, and we see that same picture for all of those. And then we test, I think this one of the ones where there's no effect, and indeed there's no effect experimentally that you see. So essentially, you can go all the way from a variant predicting its effect, and then verifying it in the particular cell type. Now, we're in a medical school, so what about phenotypes, right? So this is all great. I'm spewing out biochemistry, and for some of you, it's probably very interesting, but for others of you might not be at all. So what does it actually mean to phenotype? So it turns out that these non-coding variants, right? So we're talking about patients that have perfectly functional genes, but where there are these variants that are affecting either their expression or splicing. So in this case, expression, and they can affect survival quite significant. So this is clear cell renal cell carcinoma from the, I believe this is from TCGA. It's from one of the big cancer cohorts. I can bring it. It's either ICGC or TCGA, you know, if you are very interested. But you can see that there's a significant effect on survival of these patients based on whether they have a high impact non-coding tumor variant or not. And in fact, this is true across cancer. So this is pan-cancer looking at ICGC. So we're literally just sorting patients based on whether they have at least one high impact disruptive, non-coding disruptive SNP that's affecting their expression, right? Or those regulatory sequence classes or not. And there's a significant difference in survival. So the patients that do have these non-coding variants have a significantly worse survival. And this is not actually explained by sort of the obvious things like tumor mutation burden, which would be, you know, one obvious hypothesis, or length of genes, or any of the obvious things that we'll look at. Right. So this is actually, so these variants, so it's not just a story about the fact that deep learning models can be helpful in interpreting the genome, but it's also the story of the fact that we really need to start looking beyond genes if you're thinking about clinically relevant variants and understanding human disease. And I would say it's even perhaps, and now I'm speculating, but I would say perhaps it's even more important in kidney disease than many others, right? Because we have had less of, you know, much more of a disease heterogeneity, and that, you know, makes you think that maybe there's actually a lot of regulation that's not just in the coding regions. All right. Which brings me to kidney disease. So QRGN, I think probably, I think in the division of nephrology, many know what QRGN is, but this is a medicine grand route. So let me tell you about the study. So can we actually apply all of this to QRGN? Now, the last story was actually unpublished. It's in review. This is completely unpublished, and very much excited about all of our collaborators within the QRGN consortium. So a lot of credit goes to them for generating the data, and, you know, we're interacting with a lot to verify this. So this is hot off the press. QRGN is a multi-center consortium. It has pediatric and adult cases, and it's includes minimum change disease, FSGS, membranous nephropathy, and IgA nephropathy. It's about 2,600 cases. And very ethnically diverse. So we're going to focus on the whole genomes, right? So can we look with these methods at the variants in QRGN? And so one of the first things that we did is actually apply the method that I just told you about, right? The one that the one that predicted expression disruption based on variants, right? So if we apply, and we actually focused on, is this blocking you guys? Oh, never mind. Good. It's only blocking my view. Okay. PLA2R1 is, of course, is a protein that's quite important, and, you know, in membranous nephropathy. And that's the one that we actually focused on to show you. So this is a lot of going on in this graph. I apologize. This is so hot off the press that I couldn't get my student in time to give me a build of this. So imagine that you're only sitting in the yellow first. So the yellow is, if we go through computationally and just change every possible position to every other, right? Like say at this position in the promoter of PLA2R1, there's an A in the, you know, reference genome. We just change it to a T or a C or a G. And that gives us all possible variants, right? So this is sort of, this is ground state. And then we just reference it to the ones that are in areas that are conserved, right? Because if the area is not conserved, then probably this is meaningless. It's great. It's computational exercise. It's not that interesting. But so think of it as the background. It's the grounding of how a rare variant could affect expression of PLA2R1. But those variants are not in QRGN necessarily, right? So this is just giving you, so, you know, the really high ones are the highest possible effect you could have. So it's just calibration. Now, the blue variants are actually GWAS SNPs, right? So these are common SNPs. Most of them are not going to have a huge effect, right? Because they're common. If they're affecting this important protein in big ways, the vast majority of them, like, then we're really all as humans not in a good position, right? I mean, really, right? Like, realistically, most common SNPs aren't going to have huge expression effects, right? Because there's so many SNPs that are happening in non-coding regions, there should be huge selection to not do this, right? But look at this guy, this blue star. So this C2T variant is actually in our QRGN cohort in multiple patients, and it actually has a pretty significant effect on expression of PLA2R1. So we're looking into that variant further, right? So this is the sort of analysis that you can really do with this. But you can actually go further. So remember early on, I actually told you, well, it also matters what's happening post-transcriptionally. This is, with splicing, with transport of RNA, with stability. And this is happening this is facilitated by proteins that are called RNA binding proteins. So, right, these are the proteins that are actually splicing an RNA that are transporting it, right? So there's some really famous RNA binding proteins. If you've, FMRP, probably if you're at all looked at neurology or anything with mental health disorders, neurodevelopmental, right? FMRP is a big, I guess star is not the perfect word, but so this is coming from patients where FMRP itself is mutated, right? Or any of the other RNA binding proteins. But what we're looking at is, what if the RNA binding proteins are perfectly functional, but the binding site that protein needs to be able to process RNA for your gene of interest, or let's say a gene that's really important for appropriate glomerular function, right? What if that binding site is mutated just for that gene? So that's what we're looking at. And of course, as you can imagine, there's actually a lot more opportunities in the genome for those mutations, just because there are many binding sites and only one FMRP sequence. So we have another deep learning model that actually is trained on, for those of you interested in RBPs, on ClipSeq data. So these are in vitro models of actually how data sets that are of how RBPs are functioning. So we can actually predict the effect of any variant. So now we still need whole genomes, but this is actually within the sequence of the protein, right? But in the introns. And it turns out when we look at QRGN, those variants are actually enriched for post-transcriptional effects, right? So if you look at variants in healthy population, NOMAD is just, a big data set of whole genomes of people who are healthy, or at least, approximately healthy. If you look at all variants, same thing, they're about this level, right? Like, so we're normalizing to them. If you look at basically variants that are affecting splicing in the QRGN cohort of genes that have no association with these diseases, again, you don't see a signal. The moment you start looking at genes that have been associated with any of the QRGN disorders broadly, right, through GWAS or through prior information, here's a very significant signal, right? So there is a clear signal that variants in QRGN patients that are disrupting this post-transcriptional, right, like processing of their RNAs, are actually affecting these genes specifically. And I'll just show you one specific one. So we actually got really interested in BAF3, and this is looking at IgAN. So there's about 67 million QRGN variants overall, right? Like if you look across all patients that we have from QRGN, and we call all variants, and then we also actually threw in the GWAS variants as well, and then we limited it to genes that were expressed in the kidney, right? Because, well, for obvious reasons, right? Then actually, BAF3 is one of the ones that we found. There's a variant there that's affecting binding of SRSF1, which is an RNA binding protein that affects splicing. And so that variant is actually specific to a number of patients in IgAN. And BAF3 actually has been associated previously by other publications, including one from the Curiel group in Colombia, who is also a QRGN group, with IgAN, but not that specific variant, just the gene. So essentially what it looks like is it gives a specific mechanism of how these variants might be affecting it. So in the intron, there's this variant. Now the RBP isn't binding, the intron isn't spliced out, and that essentially can affect the ability of this RNA to be degraded or stabilized. And then the protein is, of course, not the same functionally because it has this extra intron and is affecting VAV3 function, right? So it's essentially this extra VAV3 intron that is being kept in by the variant. All right, so give me, in my 4 minutes left, I'm just going to really quickly tell you, so what can we actually go beyond these single variants and think about diseases and network effects issue, right? Because of course, this is all great, but I so far have been focusing on one gene, one variant, but we all know that while that is sometimes how we look at disease, in reality, we're looking at this very complex network within particular cell types that's being disrupted. And we need to link that to actual specific biological, biochemical, organismal, and then eventually actual clinical phenotypes. And so what a lot of the time actually that my lab spans that I won't have time to tell you about is how do we map these networks in specific cell types and tissues and understand network changes. And this is largely from a different type of data, right? So we're talking using expression data, physical interaction data, proteomics, et cetera. We're starting to look at it actually at metabolic networks as well. But how can we look at what do these, cellular networks look like within particular cell types? How do they change? So if we're looking at the podocytes, how is it different than the cellular networks in other cell type, let's say tubules? But also, how is the podocyte network changing as the disease progresses for patients? Can we identify parts of the network that are actually changing or predictive of clinical outcomes for patients with the same disease, where there's outcome heterogeneity, et cetera? So I won't have time to tell you, I'm just going to skip this, but we make these networks actually out of huge collections of data. So think of it as trying to generate a much more complete picture of the cellular pathways you've probably all seen in your genetics and other textbooks, but actually understanding this based on all the data we have now and in a very specific cell type, development, disease, progression context. I'll just skip that. I'm happy to answer that in questions, and they work. So you can actually use these networks to predict highly cell lineage specific effects. This is actually looking at cytokine IL-1 beta in the blood vessel network, and you can actually predict based on its interactors. And then our collaborators tested this, what are the top neighbors of IL-1 beta, and it turns out specifically in blood vessel, if you perturb IL-1 beta, the genes that are perturbed transcriptionally at the very first time point are significantly the ones that we've actually predicted, and actually the two that don't change transcriptionally are known interactors of IL-1 beta that are not transcriptionally regulated. Anyway, what can we do with these networks? So one of the really powerful ways to use these networks is to identify genes that are potentially like other genes that you're interested in. Let's say you have drug targets. The obvious way we usually look at drug targets is structure, right? But can we also look at the pathways, right? Like pathway membership and functionality that these guys are hitting, and try to understand, for example, off-target effects. Maybe they're not actually off-target effects, they're wrong cell type effects, right? So understanding things like that. But another one is actually trying to understand functional effects of various perturbations. So when COVID hit, we were collaborating very closely on this. Of course, we have huge number of collaboration with Matthias Kletzer's lab at University of Michigan. And so this is actually looking at the Pima Indian cohort that is also in collaboration. I'm forgetting where Rob is blinking. The Pima Indian cohort. Yeah. So basically we have this cohort data, right? So remember, actually, I'm sure the nephrologists remember, there was a huge panic about blood pressure medications. Actually, all of you would probably remember, right? With ACE2 inhibitors, right? So there was actually a critical question of, should we take patients off their blood pressure medications, because otherwise they would be killed by COVID. They would be at much higher risk of dying from COVID. And so we wanted to address this question, but of course we didn't have that much COVID specific data at the time. What we did have is this amazing cohort where we had cell type specific, so expression for diabetic nephropathy patients. So this is looking at patients with diabetes who are, of course, at much higher risk of negative outcomes from COVID, and looking at what do their kidneys look like, right? So you can look at their single cell and understand what their kidneys look like. And then you can, and we're actually specifically looking at the cell type. One of the aspects we actually identified which particular cell type was the one that that SARS-CoV-2 was actually affecting by looking at the cell types in the kidney that had a high expression of ACE2. But then also, we actually had patients in the ICU who had severe COVID infections, and we had isolated their kidney cells from their urine. And what you're looking at is actually on the left side is these patients' expression, single cell expression for these few ICU patients. So these don't have any kidney disease, right? But these patients have SARS-CoV-2 and severe COVID-19. And you can see this clear viral signature. So the key part of the networks is that it allows you to really zero in to what are the signatures, what are the pathways that are being affected. The surprising part is when we look at this at the patients on the right. So these are the DKD Pima Indian cohort patients. This data was collected before SARS-CoV-2 was anywhere near humans or existed, right? And yet you see the same signature. So essentially that gives you a very specific hypothesis that DKD PTEC cells, so PTEC cells were the ones that specifically looked like they were being impacted by SARS-CoV-2, are essentially in this prime state of essentially inflammation and viral type of signature that makes the kidney potentially especially vulnerable to adverse outcomes in these diabetic kidney patients. And of course, that means that you shouldn't take well, as we all now know, we shouldn't take patients off of their blood pressure medications for this. And so you can actually download, you can do this sort of analysis, so this functional module discovery, as well as look at all of our networks, download them if you're a bioinformatician or you have bioinformaticians in your group. At this hb.flatironinstitute.org, actually the easiest one is humanbase.io is another website. And then you can actually look or predict variant effects for any of your own sequences, or you can look for the ones that we've looked at and predict gene expression effects. This is actually tissue-specific gene expression prediction, not cell type specific ones. If you're interested in that, just let me know, because that paper is still in review, but I'm happy to give you the website for the newest one. And that's it. I didn't actually do any of this. I just go around and give talks and sometimes run grants and talk to really cool people and have great dinners. But I have an incredible group. I think I had everyone highlighted as we went through. We have amazing collaborators through the KPMP, QRGN, Neptune and other projects. And, Matthias Kretzer and his large group, Laura Mariani, et cetera, they have been incredible. And we interact with them very closely, all of this. Thank you.
Thank you. Thank you, Dr. Troyanskaya. That was a very inspiring talk. And I really liked the last part where you brought home the concept of how we can use this in a practical way in terms of understanding, especially in light of SARS-CoV-2, and infer from that what we need to do in terms of treatment or what alterations in treatment we might make. I was wondering whether or not you were able to develop the AI or the modeling for looking at epigenetic effects as well as pure genetic effects. I don't mean post-translation. I mean things that from maternal to fetal involvement that might influence what the genome might tell us, but what the epigenetic component tells us.
Yeah, that's a really great question. So we are looking sort of the closest we're going to looking at that right now is we're looking at methylation clocks and whether we can actually look at methylation effects. So that's not the maternal fetal, but it's trying to understand basically the encoding. And it looks to basically we're expanding that same method, the first method I've talked about, to actually include methylation data as well, and try to then understand if we can actually start identifying variants that might have that effect. And then eventually, so for the maternal fetal, I have a huge amount of interest in that. But to really directly answer that, we need cohorts where we actually have some of the clinical data. So if there's anyone who is interested, this would be really cool to look at.
Dr. Punjabi, come to the front so people on the Zoom can hear your erudite question.
Perhaps a very simple methodological questions. These deep learning algorithms are very data hungry. How do you know a specific network that you constructed is the truth? The second part to that is, how are these deep learning algorithms better or the equivalent or perhaps inferior, perhaps even inferior to parametric approaches? You know of many of them. I just thought if you could comment on both of those issues.
Those are great questions. So first, actually, let me answer the second question first. So I am by no means the person who, my lab, I call it, I'm not a hammer lab. Hammer labs, and there's no disrespect, but there's certain people who they trained in deep learning or they trained in Bayesian statistics, and then they will look for problems that fit Bayesian statistics. We will do whatever it takes. I mean, if the best way to do it is something super boring like correlation, we will do, we want to solve the clinical problems basically. And so I actually think for a lot of problems in biology, deep learning methods make no sense, or at least at the current state of biology. They do actually, for the problems specifically for this genome interpretation, they have proven quite superior to basically anything else that we've been able to do, purely because of their ability to really capture all of these very weak signals in the genome, right? And not rely on the supervised setup where you actually have to have either parametric assumptions or supervised setup where you are associating a genome with specific outcome, right? Because as soon as you have the supervised setup, it's great and powerful for certain things, but you're only learning about the common variants, right? And we want to be able to essentially make predictions that are just as accurate for, essentially what you're doing is the same as text to speech, right? You're just trying to understand how to read the genome. And so for that, they've actually been very powerful. People have actually tried very briefly, very early on, supervised approaches like an SVM-based approach. And there's a way you can actually finagle it to work it, but it's not nearly as accurate or as powerful. But with that, and that's why I want to answer that first, and then your first question second. But of course, with that comes a huge, I'll say, responsibility in that deep learners, of course, as you I have a feeling know, overtrain very easily, right? So you can overfit to something that's, basically to noise in your data. And you'll get these great predictions. And in biology, you have to be extra careful, right? Because you wouldn't know that it's noise because it's this very complex system. So it might not even be noise. It might be some other biological process that's actually irrelevant to what you're trying to study. So we are, basically obsessive about how we evaluate them. So one thing we standardly do is we always leave 1 or 2 chromosomes out of the entire, so our models have no idea that these, I can't remember what the chromosomes, but typical non-sex, typical chromosomes of the entire process. So they never see any data associated with those chromosomes. So the first thing we do is once it's completely trained, then we will apply it to these chromosomes to see how it does. We always use external validation, right? So one of the things you can see is, for example, this is common variants, but how do we do on GWAS variants, right? Like associated with disease. So other tasks, right? That the deep learners have no idea of disease, right? They're just trying to learn regulation. So how does it predict disease variants? How does it predict, EQTLs or, splicing QTLs, things like that. So we do all of that. If there's experimental data sets, like for example, there's digital footprinting data sets that we never use them and we will use them we never use them for training, we'll use them for experiments. And then finally, we do verify actually quite a few of our, subsets of high impact variants experimentally, as well as ones that we say are low impact. So I didn't show because of timing and, because Department of Medicine and not psychiatry, we have a pretty large study, for example, on autism, where we actually verified about 45 of the high impact variants, some of them very close to the genes, some far, all neuron expressed genes, but some genes that have never been implicated in an ASD before. And in tissue culture, 94% of them, so I think it's all but one, do cause specific effect in neuroblastoma cells on expression, just that one single letter had change. So we do a lot of verification approaches to make sure that our models work before we actually ever go further with any of this.
There's a fine line of distinction between medicine and psychiatry, so don't poo-poo that. I want to thank our Chief of Nephrology and Hypertension, Dr. Alessia Fornoni, and her entire division, Dr. Roth, for allowing this named lecture, and thank you for inviting such an inspiring speaker. Everyone who's on the Zoom, please note the address that was published for the MOC credit and CME credit. And those of you that are here, as you leave the auditorium, there's a QR code that you can use to access your CME and MOC credits. Everyone, thank you so much for coming. Happy New Year. See you next week.